So for the next part of my talk, as I mentioned, we're going to be doing a quiz just via something called, let me just move my slide on here, Poll Everywhere. So the link that one member of the team or group should go to is on my slide at the moment. So if you could just type that into your search bars, then I'll give you 5 minutes just to make sure that you have access to that. Okay, so can I just have everyone's attention again? Can I just check that all the groups have access to Poll Everywhere and have been able to submit their answers for this first question here? I'm not able to see anyone, so if you do have a comment, then please just feel free to come up to the microphone and interrupt or make yourself heard.
Did respond?
Yes, everyone answered the question, the first question.
Excellent. So the first question here is, what is your overall impression of the quality of paper 1? So that when we're talking about paper 1, we're talking about Bravo et al. 2011. And I can see that we have a spread of results where a few have said high quality, some have said medium quality, and the majority have said that this paper is of low quality. So I think there's many ways that we can think about quality. One of the ways that I would think about quality is to think about the internal validity of a study. And the next few questions can help us go into more detail about the internal validity. We can also, I'll leave a slide at the end where people can just come up and put any additional comments in, anything that they've observed that we've not asked a question about, but we'll just go through the questions one by one and we can talk about the answers as we're going through. So, do the authors report blinded assessment of outcome, Bravo et al. 2011? And I ask you to submit your answers for that, please. Okay, I can see from this question that the majority have selected yes. So in this paper, the methods and materials are actually in the supporting information. And we can see that under treatments and sacrifice, the experimenters were blinded to the treatment of each animal. But also at other areas of the methods, we can see in situ hybridization experiments, treatments of each animal were blinded to the experimenter to prevent bias in the analysis. And also in elevated plus maze, behavioural determinations were made by an observer blind to the experiment following conventional parameters. And finally, in the forced swim test, we can see a well-trained observer blinded to the treatment group scored these videotapes for the duration of mouse immobility. So we can see that there's many different outcomes measured in this paper, and some of those have been reported as being blinded. So this is quite thorough reporting for this measure to reduce the risk of bias. There might be some that I've missed, so I'm happy for anyone to jump in if there's anything that I've not noticed in the methods there. For the next question, can I ask you, do the authors report randomization to experimental group? So I can see from this that we have some answered yes and some answered no. From my reading of the supporting information, I don't think I've noted that they reported random allocation to group, but I'm happy for anyone to correct me if there was somewhere where it was stated in the manuscript. So I think the answer to this one is no, that they did not report randomization to group. So we'll go on to the next question. Do the authors report allocation concealment, so blinded conduct of experiment? So for this one, I can see there's a bit of a split again, although it's changing as I'm talking. I'll leave that for 30 seconds. Yeah, so we have the majority have said yes, that the authors report allocation concealment, blinded conduct of experiment. So when we describe allocation concealment, we're talking about concealing from at every stage of the experiment. So although blinding has been reported at many of the outcome measure sections of the methods, to be able to say yes for this question, we would be looking for people to say that even those handling the animals in the animal house, those feeding the animals, changing the bedding of the animals, would all be unaware of the group of which the animals were allocated. So blinded conduct throughout the entire experiment, from the housing to the outcome measure to when we actually take the tissues. So I think in this case then, for a CAMARADES style question, we would actually say no. So those that said no to this, I would say was correct. For the next question, do the authors report the use of a sample size calculation? So I can see for this that all participants have said no, and I believe that's the case. I don't see anything reported in the methods about how they decided on their group numbers. We do have group numbers reported, which is a positive, but they haven't stated that they've used a sample size calculation. Do the authors report pre-specified exclusion criteria? Again, this seems to be unanimous no, and I would agree that that's the case. I don't see anything about why they would have excluded animals, or if they did exclude animals for any reason. And again, do the authors report animals excluded from analysis? Okay, I can see that the majority have said no, but some have observed that they have reported animals excluded from analysis. It's not something that I've highlighted in my method, so I'd be interested if anyone wants to give us an example of where that's the case.
Hello?
Hi there.
Hello? We found that when they assessed vagotomy,
Yes.
in the end of the paragraph, they said that some of them were excluded, only vagotomized animals which did not show a significant decrease in their food intake were administered CCK, were used in the study.
Excellent. Yes, so they've said that those, they haven't actually said how many, which would have been useful, but they have said that only those that showed a significant decrease. Yeah, that's excellent actually. So they have at least mentioned
and then we thought that they were 4 because before in where it says animals, it says that all in all animals are 36.
Yep.
But then in the next paragraph we have only 32. So we missed 4 mice.
We've lost.
And we thought that those 4 mice were these ones.
That could be the case, yeah. So I think it would be great if they showed more of a breakdown of where those numbers went to, but we could, this could indicate that that's why those 4 are excluded from the next section. That's great. Thank you for that. We'll go on to the next question. So we went into some details about internal validity, and now just a general question. How reliable do you think the results of this study are? Highly reliable, partly reliable, or not reliable? So I can see most have went for partly reliable. So when we're thinking about reliability, some have said not reliable, because a big part of reliability is about how internally valid your study is, so how much confidence we can have in this cause-effect relationship. And because they've failed to randomise and they've failed to do a sample size calculations, I think this could make you question the internal validity of a study. But other things that can contribute to reliability include whether people tell you whether the stats they intended to use were decided in advance, because then we can be certain that they didn't run lots of stats tests and just use the one that supported their hypothesis. But also another thing is pre-registering your study and having a protocol available. So I don't see any reference in their methods to that kind of thing. So I would say yes, like this is a very subjective question, but partly reliable could be a good answer for this, because we know that they have taken measures to blind, they have reported why they've excluded some animals, and there are many good reporting standards here, but there's just some things missing that we would look for to be able to say it's highly reliable. So that's great. We'll go on to the next question. The next question is, how generalizable do you think the results of the study are? So when we're talking about generalizable, we're talking about generalizable between different species, different labs, and also whether it can be generalized from animals to humans. So I can see there that we've got 100% have said that there's not generalizable. I'll be interested to hear what your thoughts are on this. One thing that springs to mind for me is that the study only reports using adult male mice, so we don't know whether it would work in females, for example. We don't know whether the same results would be obtained across different species. And because it's only a set age, so they're quite young animals, 10 to 11 weeks, we don't know how it would be in older mice. So those are some things that would maybe be good to have some variability in, so that we can think that these might be generalizable to other species or other types of animals. So does anyone have anything to add at that point? Okay, so I think those are the questions for our first paper. So we'll move on to the second paper, and we can have some more general discussion at the end. The second paper is the Pradillo et al. Paper. Again, there's supplementary materials, but can we first ask, what is your overall impression of the quality of Pradillo et al. 2017? Great. I can see that the majority have said it's high quality, with a few with medium quality. Again, this is a subjective thing, so if we just look at more the internal validity to start with, we can have a think about what gives us that impression. First of all, do the authors report blinded assessment of outcome in this paper? I can see there's 100%. So this paper is interesting in that the materials and methods are actually part of the main manuscript, which I think is quite an important thing if we're thinking about experimental design, and how important these things are to have it in your main manuscript so that everyone reads it. We can see that the animals were randomized, but also assessments were performed in a blinded manner. So that's in the treatment section. And we can see for stroke outcome, neurological status was assessed blinded to drug treatment. So yeah, those are two examples of where, also in the optical density and cell quantification section, we know that all images were acquired in a blinded manner. So that's correct. So I'll just clear this. Back. Result. Not sure if that was from a previous time or this time. So, do the authors report randomization to experimental group? Okay, again, I can see that we've got 100% of participants have said yes, and that's the case. So, animals were randomized for all experiments and assessments. That's said at the treatment section. For the next one, do the authors report allocation concealment? So that's blinded conduct of the experiment. So for this question, I can see that majority have said yes to this question. We know that the assessments were definitely done blinded, so animals and assessments were performed in a blinded manner, and then we've got different outcomes have been done in a blinded manner, but we don't know that those handling the animals throughout the entire experiment were blinded. So I would say no for this one. Do the authors report the use of a sample size calculation? Yep, in this one we can see that in the treatment section, group sizes were determined by power calculations, and they've got the alpha of 0.05 and beta of 0.2. So we do know that they used a power calculation or a sample size calculation to determine their sample size. Do the authors report pre-specified exclusion criteria? We've got a bit of a split on this question. So under the focal cerebral ischemia section, we can see animals were excluded from statistical analysis based on a priori exclusion criteria, namely, if animals experienced brain hemorrhage, lack of reperfusion, or if the surgery took longer than 45 minutes and there was excessive bleeding. And they actually give us the details that 2 aged leans were excluded in total. So we know that they set up in advance these exclusion criteria and that 2 animals had to be excluded for these reasons. So I think that's thorough reporting for that section. Next, do the authors report animals excluded from analysis? We've already answered that actually, so we'll move on. How reliable do you think the results of the study are? Great. I can see that the majority have said partly reliable, and I think that's fair. We know that they've taken many measures to try and ensure internal validity. There are things that they could do to assure us that things were set out in advance. So one of those things would be to pre-register their study, which is becoming more common, or at least have a protocol that was made available after their results are published, and refer to that protocol so we know what they set out to do in advance. And perhaps also that they had set out the statistical analysis that they would do. So those are all things that could make us go towards highly reliable, but I would say partly reliable is a good assessment. And finally, how generalizable do you think the results of the study are? So I can see that everyone actually has said not generalizable. I think this is very hard to determine because we're looking at animals and there's many things that it could be generalized to. I would agree that it's probably not generalizable, but one thing that is a positive from this study is that they used both Western rats and also they also used. So there's 2 different species of rats, strains of rats, sorry. So although it's all done in rats, there's 2 strains have been used. But similarly to the other study, we're looking at experiments with young male lean rats, so no comorbidities or anything that would make us think that it could be generalized to a patient stroke population. So those are all things that would possibly be in the next stage of research, where if you find something to work in your young adult male rats, you might test it in animals with comorbidities or females, or so these are all things that could be incorporated to make it more generalizable. Thanks for your answers there. I've just left a slide here to talk about for anyone to come up if they've got further comments on these manuscripts. I know you had a bit of time to have a look at them, so if anyone wants to make any other comments on the critical appraisal of these manuscripts, then please come up to the microphone now.
There are no comments.
Okay, thank you very much. That's fine. It's hard to know how much of a pause to leave to give people time to comment. Thank you. So just to summarise for the final 5 minutes, we have covered a lot in this lecture about different measures that we can take to try and increase the reproducibility of science research. So if we think about how scientists might approach their projects, we can think about a few different areas. So we can think about which parts of an experiment are exploratory and which parts are hypothesis testing, because we can do this type of research in very different ways. So exploratory might not require a sample size calculation, and it might not require you to set out in advance what your hypothesis is and your stats that you're going to use, but it can be used as your starting step to then go on to use hypothesis testing, where you would test using a p-value. So these are things that you can set out when you're writing up your research on whether it was exploratory or hypothesis testing. And it's important to think about what the community standards in a field are, and it's definitely becoming more common that people are trying to adhere to these reporting guidelines. So within vivo studies, people are thinking about the ARRIVE guidelines, or at least the individual publisher guidelines. And importantly, can you randomise, can you blind during the entire experiment and also during just outcome assessment? And when we are testing a hypothesis, it would be important to assert your statistical analysis plan, cement your protocol where it can be checked, and that could be shown in advance, or it could only become available once you've completed your experiment. Also describe in advance any criteria for rejecting the null hypothesis, and also make sure you have adequate statistical power to deliver a reasonable positive predictive value. So this means that we would have best practice with internally valid studies, and it could iterate through improving the robustness of evidence. So not every study needs to use comorbid animals, but we do need many models and outcomes with construct validity. And we need to understand the range and quality of evidence. So raw data that is fair would be really useful. And we want to do confirmatory studies, clinical trial tests, and use clinical trials to test interventions under conditions where effectiveness has been demonstrated. That's how we can improve what we're doing. If we think about the very best practice, that would include doing a systematic review of existing knowledge before you do your experiments. And this means critical appraisal of findings supporting your premise. And before you start your experiment, make sure you know your study population, hypothesis, intervention. This can all be set out in your protocol. Your statistical analysis plan, what primary outcome measure you plan to use, and the details of your sample size calculation. It doesn't have to be a published protocol, as I said. It could just become available. You could put it on Open Science Framework to only become available once you publish your study. And it should be somewhere with a persisting, unique digital identifier. You have to think carefully about the experimental design, and that could be using something such as the NC3R's Experimental Design Assistant. And the other option is to do registered reports or registered funding, where you submit your protocol, you get feedback, peer review feedback on that protocol, and then they'll say that they will publish your results regardless of whether it supports or rejects the null hypothesis. And that's an interesting way forward for how we could be publishing our results. Just for the final very best practice, we would want to, during the experiment, manage our risks of bias, look at the traceability of all the materials we use, make sure we keep records of everything that we've done, and write standard operating procedures for most tasks. We should be aware of pre-existing and emerging compounds and record any deviations from the study protocol. And as I've mentioned, after the experiment, make that data and code available, make the preprint available, for example, on bioRxiv before you publish, and try and not chase impact factors, but make sure that that data gets out there, even if it's not in the highest impact factor journal. And just as a conclusion slide, we would want to think about methods to reduce bias, make sure we're using the appropriate number of animals and appropriate statistical analysis. So thank you very much for your time today and for taking part in that practical exercise. If you've got any questions, then please just come up to the microphone.